classify a mutant or a variant as variant of concern? When there is sufficient evidence to say that there is increased transmissibility. That means in public health, it's very important. The same virus can infect more people compared to the earlier variant or the earlier parent virus. Or when there is evidence to say that the virus is more virulent, which means its clinical phenotype is different and it kills more people. And also evidence to say that there is decreased effectiveness in the public health measures that are routinely adopted. A variant of interest, on the other hand, is something that is spreading, but we still do not have both cell culture evidence, laboratory evidence, and public health evidence to say that it's more infectious. So it is put on a hold till data is collected. Now, let me tell you that before I go on to tell you what the variants we have today, they must tell you something happened in end of February. Italy reported one of the most important mutations in the spike protein, which is what classified A to B, and it was called B.1 at that stage. Now, this was a very significant mutation resulting in a change in an amino acid at position 614 on the surface of the virus in the spike protein from D, which is aspartic acid, to G, that was glycine. Now, this particular mutation made the virus more infectious. And you can see on the graph on the top left panel, the D614G mutation completely took over somewhere from the end of February, and by May, it had completely replaced the parent Wuhan virus across the globe. So it was more infectious. Why I'm telling you is this is the grandmother mutation. From this, all other mutations that came have been on this backbone where they definitely have this grandmother mutation. Because of the confusion prevailing in the terminology of B.1.1.7, 351, P1, and so on, the WHO about 6 to 8 weeks back decided to give simple names, Greek alphabets, alpha, beta, gamma, and delta. These are variants of concern. That means there is evidence to say that they spread much more than the parent Wuhan virus. And then epsilon, zeta, eta, theta, iota, kappa, and lambda. Lambda is in the news, all of you know, in the last 3-4 days. So all these are variants of interest. That means they have been reported from multiple countries, but they have not spread globally. They are spreading rapidly. So they have been put in the category of variants of interest. Whereas alpha, beta, gamma, and delta, alpha was the one first reported in UK, which crossed a huge surge in UK in December. So this was called the alpha variant. Beta was reported in South Africa, and that was reported in December. But as early as May, people had noticed these mutations. Gamma was from Brazil, reported in January, and we in our country reported Delta as early as April 2021. All the others I have listed here, I'm not going to read. And the latest one, of course, in the last row is Lambda, which is doing a lot of rounds in the region. On the right, I have given you the names of the variants, the original pangolin lineage, and from which country it came. Okay, these are the spike gene mutations in alpha, beta, gamma. They are signature mutations. The alpha, the signature mutation is N501Y in the spike protein. Beta is That mutation plus E484K, and these are amino acid positions, and I'm talking of spike amino acid positions. And when we do sequencing, we look for these mutations and then do variant calling. Gamma is 501Y. That means it has the same UK variant and South Africa variant, and it also has the same South African variant plus additional mutations. Delta is the original grandmother. All of them incidentally have original grandmother mutation. And the signature mutation is in position 415, for a lysine is converted to arginine. Okay, so what? So what? Viruses mutate. We know that. What's the big deal? Why are we talking so much and why are we making such a big noise? Because it has implications for transmissibility, it has implications for virulence, and it has implications for antigenicity. If you look at transmissibility, all the 4 variants of concern are highly transmissible. The UK variant was 50% higher than Wuhan. Then the South African was also 50% higher than Wuhan. The Brazilian one was also 161% more. And the most infectious is the Delta variant. In terms of virulence, we have only 1 or 2 where the virus is supposed to be more lethal. One study, a couple of studies that were reproduced in UK saying that maybe clinical phenotype is more, and so also with the Brazilian variant, but not with the other variants. We don't have concrete data. In terms of antigenicity, the biggest culprit is the South African or the beta variant, which has shown the maximum escape from existing vaccines. When I say maximum escape, I must qualify saying that all vaccines protect against all variants when it comes to serious disease and death. But in terms of infection prevention, the South African is the most resistant. The Delta Plus made rounds about 3-4 weeks back. Huge noise, huge. It came with a bang and it's gone with a whimper. In my opinion, it's a misnomer. It should be called Delta minus, because all samples that were detected positive for Delta plus in India were samples collected in April end and in May 15th or so. If it had been more infectious than Delta, it should have caused a huge surge. It has not caused a huge surge in any part of the world. So people believe that it is not a major variant of concern. WHO has in fact downgraded it to variant of interest. Okay, how do we detect COVID variants? It is by sequencing. As you all know, India has formed a consortium of genomic labs. It's called Indian SARS-CoV2 Genomics Consortium. It is supported by Department of Health and Department of Biotechnology. And the main reason it is being established is to look for variants, look for new variants, as well as to look for associations of variants with increased transmissibility. Close to 50,000 samples have already been processed, and 45,000 as of today have been sequenced. And remember, the contribution of INSACOG is to the world has been demonstration of Delta plus variant. Basically, what happens is samples with a CT value less than 25 are sent to genomic labs. In Karnataka, we have NIMHANS and NCBS. We are very soon adding IISc and JNCASR. Any sample that has a CT value less than 25, the sample RNA is extracted, converted into DNA, and then put through a sequencing machine. The whole process takes about 3 to 4 days, but then it throws out so much of data, because when you do sequencing, you're not doing only viral genome sequencing, You're also having host genome sequencing. So to wade through that data and then identify SARS-CoV-2 genome and then match it to existing variants and then do a variant calling, it takes about a week. So overall, from the time a sample arrives to the lab, it will require minimum of 8 to 10 days before a report is generated. Point number two, sequencing is very expensive. It cannot be done in all RT-PCR labs. The most important requirement for sequencing is expertise in interpreting the large amount of data that is generated. So it is exacting and it requires expertise. Indian INSACOG has followed the WHO guidance for doing genomic surveillance. A total of 15 samples per site every 15 days will be sent in every state. How many sites? 10 sites. What are these sites? 5 hospitals and 5 labs. And what are the categories of samples that are chosen for sequencing? It cannot be routine samples. International travelers, of course, because we import variants. Representative samples from clusters and focal outbreaks. Cases of COVID-19 reinfection. Infections who've been in individuals who have received 2 doses of vaccine and completed 2 weeks, which we call breakthrough infections. Any cluster or group referred by you people with severe morbidity or mortality, and then of course any of the pediatric COVID-19 cases where there is some interest because we want to know if there is a mutant affecting pediatric children. So these are the only categories. Not every routine sample that is PCR positive goes into sequencing. What are the implications for COVID-19 variants? As I said, there are 3: on transmission, on replication, and vaccine escape. Here is a map where I put together the implications for transmission. The top left one is the first one, the UK variant. It came to light because there were reports of increased cases in Kent County of UK, and within a matter of 4 to 6 weeks, UK had a huge surge in December, January. And these were thousands of cases. And that's when they identified this variant. They grew it in the lab. They showed it's more infectious in cell cultures. They also showed through public health data that it's more transmissible. In a matter of few months, you can see the map on the top right. It's a heat map. Heat map meaning darker the color, more the number of cases, lighter the color, less number of cases. You can see it had spread across the entire globe. Then we had, of course, the Delta variant, the map on the left with India having a huge red dot and spreading. It has now spread to close to 100 countries. And this is the most predominant variant that is prevalent in the globe today. And in the last 2 weeks, we had the Lambda variant. The Lambda variant was first detected in Peru and now then spread to Chile and Argentina. Cases have been reported from the United States, from Europe, from Australia, and as many as 29 countries. The map on the right, that panel you see, it shows you that in Peru, it has completely replaced circulating variants and it's only Lambda that is present. In Chile, it is 30 to 40%. And in Argentina, it's raising its ugly head. It's already 20%. It is still not reported from India. Look at our own data from our country, right from December 2020 on the x-axis to 12th May, and I've actually not put the recent one. If you go up to June end, you will see this mauve colored bar increasing. That is the Delta variant. Initially, for 3 to 4 months, the indigo blue you see was a problem. UK variant was spreading extensively in Delhi and Punjab. It contributed to the surge. Then it was replaced with B.6.17.1. That died out, and then came the Delta variant. Now almost 90% to 95% of sequences being reported or the Delta variant. Then we had, of course, Delta variant, which is not more infectious and it has not replaced Delta. We certainly don't have any evidence to say it is more virulent, and we already have information that current vaccines work against Delta plus variant. So, in my opinion, misnomer, it should be called Delta minus. What are the potential consequences of emerging variants? A, faster spread. B, there is some concern that it will make miss routine diagnosis, but this has not been borne out because most diagnostic tests PCR today do not use only spike protein region. They use many regions. We do a multiplex test. We use many genes. So the chances of missing a variant is very, very less. Decrease susceptibility to therapeutic agents, yes, especially to monoclonal therapy and convalescent plasma therapy, and that too with the South African variant, the Beta variant, and some reports on definitely the Delta plus as well as the UK variant. Evasion of natural vaccine induced immunity. Actually, there is no hard evidence to say it causes serious disease in vaccinees. That is not happening. But infections are happening with the variants after vaccination. And the notorious among them is the South African variant B.1.1.